Hi, my name is Ahmad, and I've been working with the Friedman Lab this summer, looking specifically at TRIC, which is a protein chaperonin that folds up to 10 to 15% of the entire proteome, including some proteins like actin, which are vital cytoskeletal proteins. TRIC is very important because, again, it's folding so much of the proteome, which means it's implicated in a lot of folding-related diseases like Alzheimer's. And so one way to look at the mechanism of TRIC and understand it better is to compare it to a simpler archaeal ancestor known as MMCPN. And so my summer project was basically looking at how broad MMCPN's compatibility is with a set of different actin substrates. So the first thing we did was we purified MMCPN. To do this, we simply expressed it in cells, did some cell lysis, and then did some initial cleanup with ammonium sulfate. Then we did three different rounds of chromatography. So first we did a Q column, then a heparin column, and then a sucrose column. We basically separated the proteins by charge, surface interactions, and then molecular size. And you can see from figures B, C to D that MMCPN is gradually getting more concentrated and impurities are gradually getting less concentrated, which is good. And then we did some further purification for specifically the actin substrates. Very similar process. You express it in cells, you lyse the cells, then you put them in a solution to denature these proteins because we want to see if they're going to be able to be folded by MMCPN or not. And then we did his tagged chromatography, and then we got similar results. In figures F and G, you can see that the bands for crinactin, asgard actin, and human beta actin, the three actins we're interested in, are pretty highly concentrated. Okay, so now for the actual assays. The first thing that we did was we wanted to just see if MMCPN could bind to all of these different actins and how well it'd be able to bind to any of them. So we just incubated some MMCPN with three different samples of the three different actins. And then we just we ran a gel on that and we saw how highly correlated they were. And so we saw that MMCPN actually unusually had a very strong signal with human beta actin. And so it bound well with human beta actin, and not as well with the archaeal actins, which were asgard actin and crinactin. At first, we were like, okay, maybe it's just binding and it's not really gonna fold it as well. But turns out in the folding assay that we saw roughly the same exact result. Um, Now, in the folding assay, it was a little bit more complicated because MMCPN relies on ATP in order to do full folding. And so what we did was we did a control lane with no ATP, and then one with ATP, and then one with ATP ALFX, which is basically a variant of ATP which can't be hydrolyzed. And so we also shouldn't see very much folding there. So unusually, the ATP control lane in the folding assay didn't actually work. And so in the non-ATP lane, we still saw some folding, in fact, a lot of folding. And this is unusual. Even though we repeated the assay, we saw the same exact result. And so this is something we might have to resort to a number of different assays to resolve. We still have to investigate that. But regardless, the same exact pattern that we saw, where human beta actin consistently produced higher and stronger signals in the gel and in the membrane when we did the Western blotting, still showed up in the folding assay. And so now it seems like MMCPN not only binds to human beta actin better, it also folds it a lot better. And then we saw the same exact pattern again repeated with the protein kinase actin assay. So again, we have the same exact conditions as the folding assay where you have a lane that's a control lane, then an ATP lane, and then an ATP ALFX lane. The only difference here is that we also have to add protein kinase. And so protein kinase basically chops up any protein that is not properly folded. And so it's a good way to detect if your protein is properly folded or not. And so we can see that these bands right here correspond to the folded actin, and then all the bands below correspond to the cleaved actin that wasn't folded properly. The cleaved actin, there wasn't very much of it. There was much more folded actin, and again, the signal was stronger for the human beta actin relative to the two archaeal actins. Again, very unusual. So conclusions and future directions. Essentially, it seems like MMCPN might be able to fold human beta actin better than it can fold archaeal actin. But again, the control wasn't really well established, so we're going to have to do some more testing. We're probably going to have to come up with a more quantitative assay to fully flesh out this relationship. Some future directions are basically comparing MMCPN folding to trick folding, which again is the human eukaryotic version, and then perhaps even doing some mass spectrometry methods to actually look at um, perhaps some thermodynamic or structural features that could explain differences between the substrate specificity of human trick and archaeal MMCPN. And in fact, we've already purified a lot of trick in order to do this. But yeah, that was the project for this summer.